Uh, so good afternoon all. I hope I'm audible to everyone. So I, Priyanka Bhupaye, welcome you all to this webinar, Guidelines for Competitive Examination on behalf of Dr. D.Y. Patil, Biotechnology and Bioinformatics Institute, Pune. First of all, I thank you all for joining with us. I hope you all are doing good and keeping well. So the idea behind this seminar is to encourage and motivate our students to provide a platform where we can solve your doubts regarding some competitive examinations like DBT, JRF and all. Because what happens many times, we wanted to appear for some examinations, but we do not have some guidance, the proper guidance. So here we are trying to help you with some guidance. So today we have with us Professor J.K. Pal, who will give us some idea regarding this examinations, how to prepare for the examinations, and what type of questions we have to prepare, and how to prepare for the examination and all. What is, I'll tell you this first. This session is divided in 3 parts. First, we have one idea. All of us, the faculty members, decided that we should create one YouTube channel where we can provide some information to all the learners of our college and for other colleges also, so that you can refer that material. So today we have an inauguration session of our YouTube channel as well, and after that we can have a video session with Professor Pal sir, and then we'll proceed for the question and answer session. So now I'll request Professor Neelu Navani. She is a faculty at our institute, and she is a hardcore researcher as well. So I'll request her to give about the idea of this webinar, of this YouTube channel, and how it has started and all. So over to you, Neelu madam.
Thank you, Priyanka ma'am, and welcome all the students. It's a great pleasure to be here on the first webinar of our institute, and we take this opportunity to inaugurate the YouTube channel of our institute. So this idea came up because we thought there was no, other than ERP, we wanted to have an open resource portal where we can have a lot of educational material, videos and other material created originally by the teachers and also by the students of this institute. So the whole idea was to create an open resource material which will be available not only to our students but to also other students of other colleges and other institutes. So with that idea, we have already uploaded few videos on this YouTube channel, and today we would be requesting Professor J.K. Pal to inaugurate this particular channel, and we appeal all the students of this institute to subscribe to this channel. Also, you can request all the alumni, your seniors, to join this channel. We want more subscribers and we want more likes and also dislikes on the video. Professor J.K. Pal, may I request you to please inaugurate the channel? So you're
to inaugurate the YouTube channel, which our faculty members have put efforts to put together. And as Dr. Navani has explained, this channel is going to be another platform where we can put up all different kinds of activities, the different videos and educational information into this channel. So it's my pleasure to inaugurate this channel, and let's see whether this is the YouTube channel. And can you see the YouTube channel?
No, sir.
Now?
You will have to probably go out from the PowerPoint and then open the browser in the share screen.
Okay. Now you can see it?
Yes, sir.
Yes.
Yes.
You can see
now it
the
is
channel.
visible. Yes.
Okay. So I hereby inaugurate the YouTube channel of Dr. DYPBBI, and I'm sure all the students and the faculty is going to enhance the strength of this in terms of providing all valuable information, which should be, of course, screened through a committee prior to putting into the YouTube. So thank you very much. Thank you, faculty and the students, for helping in connecting different kinds of movies, which already we have prepared. And to begin with, we have 4 or 5 videos that are part of the YouTube channel. Thank you.
Thank you, sir. Thank you.
All right. So now we are going to go to the main topic of the webinar today, that is guidelines for the competitive examinations, particularly for the career after the degrees that these people are completing, BTech, MTech, MSc, and so on and so forth. And one of the major being the research career. And for that, PhD program is the beginning, and then it goes on. So first part of this is going to be a recorded message.
Here I'll request all the students to write your questions by the time you can see the video. You can start writing your questions in the given link so that we can answer your queries. The questions, like, I guess there is some confusion between what type of questions which are related to the today's topic. That means what type of examination you wanted to give, you have facing any problem, what are the study materials we wanted to refer and all, such type of queries, how to proceed and all. So that type of queries you can write. Where to appear, what is the use of giving the examination and all, that type of queries we are asking you.
Dear students and colleagues, greetings from Dr. D.Y. Patil Biotechnology and Bioinformatics Institute, Pune. I'm sure all of you are coping well with the current crisis and are safe and healthy. As many of you are aware that in order to compensate for the absence of summer training program as suggested by AICTE due to COVID-19 pandemic, we, the teachers at DYPBBI, have proposed a few important activities, including review writing competition, webinars on various specific areas of biotechnology, and a workshop or webinar on guidelines for competitive examinations for higher studies. The latter is the subject of today's webinar. We plan to have discussions in 2 parts. The first one, we would concentrate on the details of the examinations of CSR and DBT, syllabus, question paper, and various strategies for a successful examination for DBT-JRF programs. And the second half, we are going to solve a few specific questions, and we will justify the reason for selecting the correct choice while discussing. At the end of it, we will have question and answer sessions where all the students would be willing to ask questions. We will be very happy to address those questions, and that's how we are going to conduct the webinar. Broadly speaking, this slide shows are the few career options for aspiring students in biotechnology. As seen, the list is topped by research, although the other three, teaching, industry job, entrepreneurship, also require research to some extent as a part of the profession. I'm sure I need not elaborate as to why research is so important. Please remember that research is not only a profession, but is a passion. It is with this passion people opt for research and eventually make discoveries and breakthroughs in different areas. Research provides you the joy of knowing the unknown, which embeds within itself creativity and innovation in science, which eventually materialize in the form of development of modern technology beneficial to mankind. One point to note here, that research in academics or academic setup deals predominantly with fundamental discoveries, while for the industry it is directed towards product development. Friends, the journey of research in India in particular is long. As seen in these slides, it starts right from the undergraduate level and goes a long way to finally establish one's niche in this area among the three options that are listed below. However, the path can be easier and shorter if one acquires the distinction of qualifying JRF exams. Otherwise, one has to prove one's credibility through long training procedures as depicted in this slide. Of course, on an average, one needs to spend about 4 to 5 years to complete a PhD program in life science in particular. In this slide, I have enlisted an array of JRF examinations conducted by various funding agencies for research. Historically, although the first JRF examination by CSIR UGC named it as NET, National Eligibility Test, the name appears to have lost its purpose, particularly because when a number of research agencies like DBT, ICMR, ICAR, DAE, etc. started their own JRF program. Although many discussions took place among the heads of the major scientific organizations to have only one JRF exam, particularly for avoiding the multiple exams hassle for the students, there wasn't any fruitful outcome of such discussions. On top of that, in the meantime, the state governments started their own examinations like SET, State Eligibility Test, for eligibility for lecturership in colleges and universities. To my mind, one national test should be adequate to determine the eligibility for both research and teaching all over the country. For a common knowledge, this slide enlists various government organizations which support research grants and research positions within them. You may be aware that the principal investigators of such funded research projects can appoint CSR EGC A-less category candidates and also DBT-JRF category 2, both of these which do not carry fellowship along, through an interview and offer fellowships from their respective research grants. Another advantage of qualifying for JRF is getting an opportunity to pursue your research in a chosen institution as your place of research, and most often one can choose his or her own guide in the area of one's interest. Listed below are some of the distinguished places for good quality research. For example, there are many research institutes like CSIR laboratories listed there, DBT laboratories, large number of them, ICMR laboratories, DAE institutes, and there are central and state universities like JNU, DU, BHU, etc. In addition to those, you also have ICERs and IITs and Indian Institute of Science and similar institutions. However, due to these diverse JRF examinations, many of these quality institutions have started their own additional test for PhD programs, including the interview for selection of their candidates. As shown here, there are other research positions are also available, like project assistants, research assistants, teaching associate in various universities and research institutions. Now, as far as your interest to do PhD abroad, there are many such opportunities. There are country-wise selection and admission procedures through various types of exams and also through online interviews, and some of them are listed here for US and various European countries, and perhaps a more detailed discussion on these can be organized at later dates. Now let's look at the other side of it. The primary objectives of recruiting institutions for the selection of JRFs are very obvious, and there are some 8 points that I have listed here. You can go through these. However, the highlighted panel on the left summarizes the key qualities that are looked for in an aspiring PhD student, like subject knowledge, analytical approach, hands-on experience, research aptitude, and orientation. In some institutes, they also look for the mathematical abilities of the students. Now coming back to our preparation front, now these are some of the basic strategies that are very, very useful for preparation for the JRF exams. Step 1, familiarity with the syllabi. specific examinations and organizations would have their specific syllabi. This is depending on which examination you would like to appear for. You need to be familiar with the syllabi. Second step, select topics that are more familiar and you are more confident on the details of the topics. Step 3, always consult standard and better textbooks for your preparations. This is important because many questions are set from the textbook chapters. Lastly, practice solving questions from at least 3 previous years. As I said, these are some of the top strategies. However, it is not exhaustive. Individual people may have more additional strategies that may be useful for preparation for this exam. This slide shows examples of some important topics in different subjects: molecular biology, cell developmental biology, immunology, and in brackets what are shown are the authors of many of these standard textbooks. For example, if you talk about molecular biology, Watson, molecular biology of the gene; Weaver, molecular biology; Alberts et al. And Lodis et al., the molecular cell biology books; Jean, Levin, and Molecular Fundamentals of Molecular Biology by Paul and Gaskarby. Similarly, other subjects, there are standard books. And in this, we show genetic engineering. There is one very prominent book, Primrose and Twyman. And there are many analytical techniques which are part of most of the syllabi of different JRF exams. So it is important to prepare from different kinds of books where most of the techniques are dealt with. Shown here at the cover page of some of the standard textbooks. Now, it is important to consult the latest editions of the books, and the easiest way to get to know about is to search in the internet, and you will definitely be able to see the latest editions of the book. The question paper for JRF exam, I would like to highlight only 2 different exams. One is CSIR UGC NET and the other one subsequently DBT-JRF BET, Biotechnology Eligibility Test. The major difference between these 2 JRF exams is CSIR UGC is for entire life sciences. Means it covers a broad spectrum of areas in life science. On the other hand, as you will see later, DBT-JRF principally deals with students who are from biotechnology and bioinformatics area. Although it is free for all to take that test, but the subjects are quite specialized. Now coming to the CSIR UGC NET exam, it's a one single test paper with multiple choice questions. There are basically 3 parts, A, B, and C, and there are questions and there are options for each of these parts. A basically is including questions from general science, quantitative reasoning and analysis and research aptitude. B and C, on the other hand, they are subject related question. The difference between B and C is that B basic questions and C more critical and analytical type of questions, and therefore the questions in the part C, each one carries 4 marks as opposed to 2 marks for Part B. One thing to note here is that for most of the MCQ-based multiple choice questions, there are negative marking, roughly around 0.25, 25% for each wrong answer would be deducted. Now coming to the syllabus, these questions are basically set up from the syllabus that are already available in the website, and highlighted here are the major topics in the life science syllabus of CSR UGC NET. Now details of topics within these major heads can be seen from the CSR website, and I am not going into the details of them. Now I was talking about the syllabus for DBT-JRF BET examination. It also has one paper, but it has two parts, Part A and Part B. Part A is going to be compulsory. There will be 50 questions and all to be answered. Part B, there are 150 questions and only 50 are to be answered, because Part B covers a wide range of specializations in biotechnology. All students may not be familiar with all of them. Therefore, only 1/3 options are adequate for scoring for this particular part. As you see, Part A is basically for, again, aptitude and general biotechnology, and Part B, where you have specialized branches in biotechnology. And some of these branches are not taught to all biotechnology disciplines. Some are very specific, like agricultural biotechnology is only taught to the agriculture MSc biotechnology in more details. MTech biotechnology for the engineering disciplines, then bioinformatics computational biology, particularly for the bioinformatics students, then marine biotechnology, there is MSc in marine biotechnology, medical biotechnology, and besides biotechnology, there are two more subjects which traditionally has been part of this curriculum: molecular and human genetics, neuroscience, and you also have pharmaceutical biotechnology. So there are some tips how to attempt the questions once you receive the question in your examination hall. First one, read the top instruction page carefully. You should not miss out the instructions. Read the question paper a number of times prior to answering. Third, first answer those about which you are sure and confident. And now start answering those of second priority. Leave it unlocked so that you can come back later and confirm them before submitting. And lastly, of course, minimize the number of answering doubtful and unsure questions because of negative marking, usual feature of MCQ type of tests. Now, what is after PhD? There are multiple career options after PhD. One can have an intermediate prior to taking up a permanent profession. It could be postdoctoral research. This can be done in India as well as abroad. And then there are various other job options. Another is teaching profession, and lastly, entrepreneurship. The various job options include being research scientist in institute, be a science manager, be an expert in the industry, or head of R&Ds, and so on and so forth. There are many postdoctoral fellowships. The postdoctoral fellowships basically gives one an independent capability of conducting research, which is not true in case of PhD, because PhD is basically a training where mostly guided by the supervisor, and you have to work on most often the problem suggested by the supervisor, which is part of the research thrust areas of the supervisor. In postdoctoral fellowship or tenure, it is more or less the same, except that you have more independent abilities that are tested out, and you become more independent researcher as opposed to while you are a PhD student. So therefore, postdoctoral fellowship is a good idea before you settle down to a permanent profession that you would choose for the rest of the life. Now, for postdoctoral fellowship in India also there are many, like D.S. Kothari postdoctoral fellowship from UGC. Then CSIR organizes postdoctoral fellowship for abroad, like Nehru Science Postdoctoral Fellowship, Fulbright Nehru Postdoctoral Fellowship, and DST has their National Postdoctoral Fellowship, and UGC also has postdoctoral fellowship for women candidates, as well as DBT has postdoctoral fellowship. Now, one more interesting thing is for women scientists in particular, there are two special programs offered by DBT called BioCare and DST called Women Scientist. Various schemes are there, and these schemes are OAS A, OAS B, OAS C, and specific purpose this kind of scientist schemes are available, and these particular schemes are mainly meant for the women who have had a break in the career for many reasons, including the family reasons. Now I think I conclude on the first part of the discussion, and now we are going to move on to the hands-on, that is solving questions from a few different examinations. And at this point in time, I will talk to you directly and then have discussions as and when required. At the end of it, we will have question and answer sessions, and I'm sure all of you are going to be benefited from these small discussions that we had, and I would wish a good going to cracking these JRF examinations of different kind. And remember, more or less the examinations are similar kind. If one can crack one, then it's probably easy to crack many others. And now I leave it to your experience as you go along. And now I'm going to start the. Well, are you all there?
Yes, sir, we can hear you.
Okay, so I have concluded the first part of the discussion. Now I'm going to go on to the next part, that is the question answer, and that also I will share and I will talk, but I'm not going to have records on that. So I will again start sharing the screen with those, and I will continuously talk while I show the slides. Okay? Or we will pause for a little while and ask if there are questions. What should we do?
We can have some questions meanwhile.
We already
Or
have
I finish.
2 questions from audience.
Or I finish it and then have questions. What do you think?
I think we can finish it and then have questions.
I think that's better. So I will share again. Okay, now can everybody hear me?
Yes, sir.
Okay, so now there are some selected questions that we are going to display and also discuss. what is the criticality in terms of putting the tick mark on the right kind of question instead of wondering which is right, which is wrong. What are the strategies that one should take to select out? Now, normally what happens, the students have to read the question very carefully and naturally the options. Now, out of 4 options, often you can eliminate, first thing, eliminate the options. And you can eliminate the options, up to 2 you can easily eliminate. And when the remaining 2, you are reading and reading, and out of these remaining 2, 1 is the one which you have to choose as the right kind of answer. So if you can use this kind of strategy, then your chance of ticking or answering the right option becomes more. Now here in the first question, there are some analytical question example are given. The strength of an acid depends on its tendency to. Now here there are 4 options, for example. Now acids, they are basically the matter of protons, So therefore these options C and D, they becomes absolutely unacceptable options. So by doing that, if you have little thinking process, you can eliminate 2 of the 4 options. Now what remains is accept protons and donate protons. Now the strength of acid depends on, it is the Arrhenius concept, which you must have studied in your chemistry in the 12th standard. So naturally, an acid is always donating protons. The strength of the acid would depend on how much it can donate. So the answer is B. Now coming to the next question. Now many of the questions which are with negativity, they have been highlighted. You have to keep that in mind. Either highlighted by bold or by underlining or sometimes in italics. Now this, if you skip while reading the question, then your answer is going to be completely wrong. So in question 2, what is the question? Which one of the following statements about lysosome is incorrect? So that is to be kept in mind. So now if you go through all the 4 options, you can eliminate one by one because they're all correct, and you then come back to the actual incorrect answer. Now here, the last option if you talk about, which you are common knowledge, it contains various degradative enzymes. Definitely it is the correct answer, a correct statement. It is present in all types of eukaryotic cells. That is a question mark. It carries out degradation of obsolete components, which is true. It is a single membrane structure, which is also true, because among all organelles, lysosome is only a single membrane structure. So that means A option cannot be incorrect, B option cannot be incorrect, D option cannot be incorrect. Now we eliminated 3. Now naturally, the C option is probably the answer, because many of the plants and the yeast, they don't have lysosome, but they have vacuoles. So what is here? That is the trick that we are applying, eliminating one by one which are obvious, and then debating one or two, and then finally choosing the right answer. Now coming to the question number 3. Which one of the following produces haploids in anther culture? Now the word that has to be kept in mind in the question is the haploid. That is the key word here. Which one of the following produces haploids? Haploids as opposed to diploids, right? 1N. Now if you keep on looking at the options, anther wall, the wall cannot be used for producing anther culture. tapetal layer cannot be because they are diploids. Connective tissue cannot be, they're also diploid. So by thinking that if you have hit upon the word haploid, automatically 3 of the options you can eliminate. Now remains the 4th option, young pollen grains. Now pollen grains, when they are young, they are haploid. So if you target young pollen grains and make anther culture, it is naturally going to be haploid. So again, what is it that is important here? Read question carefully, identify the keyword and analyze in your mind. If that is the keyword, all options when I go through, I have to keep that in mind. So answer is D. Now question number 4, which one of the following statements regarding interferons is not true? Again, not true is in bold. So while reading the options, you have to keep that in mind, not true. So you are looking for a statement which is not true. Like interferon, if you talk about comeback from the end, D, interferons have anti-tumor activity. That is true. Interferon gamma increases MHC class 1 and 2 expression in antigen presenting cells. That's true. Interferon beta is produced by cells infected with virus. That is true. And then what remains is the option A. And that is what is the answer, because interferons are produced by lymphocytes. That is not true. It is produced by many other cells, and lymphocytes generally do not produce interferons. Now coming to the question number 5. Now there are some questions which are going to be calculation-based question. Arithmetic calculation, simple arithmetic. One need not get completely switched off when you get this kind of question. These are common sense questions, simple calculation. Now here what is shown, the cell-free extract prepared from E. coli cells, overexpressing enzyme beta-glucosidase, showed the activity of 1.5 units per ml, protein concentration 2 milligram per ml. Your emphasis should be reading these two particular portions, 1.5 units and protein concentration 2 milligram per ml. The nickel NTA purified preparation showed the activity of 75 units per ml and concentration goes up to 100 microgram per ml. Now calculate the full purification achieved in the later step. Now quickly you should remember, this is a question from the protein purification protocol. There are many steps through which proteins are purified, but here it's only one step purification that has been talked about, and that is affinity purification, nickel NTA column mediated purification. Nickel NTA only would bind to the histidines, and any recombinant protein which has a his component, which is has a tag. Now they all are going to bind to this nickel NTA column, and therefore rest of the junk materials or unpurified part is going to go through, and then later on selectively you can specific conditions to break the purified protein from the nickel NTA and get the purified component diluted. And in that process, there is a full purification which is increasing. Now, if you look at 1.5 units and 2 milligram, this ratio, which has become 75 units. So how many fold enhancement that has taken place? That is 15-fold enhancement has taken place. 1.5 units, 75 units. So it is many fold concentration that has been achieved. And 2 milligram per ml was the protein concentration, which has gone down to 100 microgram per ml. So that means you have almost 50 times enhancement that has taken place, 100 microgram versus 2 milligram. So it becomes 100 microgram per ml and 2 milligram per ml. So in that process, there is about let's say, 1,000-fold purification that has been achieved. So this way you can analyze the questions and you can easily arrive at the right answer. See, here the options which were there, 0.001 fold, 0.02, these options are automatically gone. So now it is between 50 and 1,000. So using those two parameters, if you calculate, then it becomes 1,000-fold purification. And protein A from Pseudomonas putida contains 135 amino acids. The number of nucleotides present in the gene encoding the protein will be, this is simple calculation, again arithmetic. 135 amino acids can be coded by 135 into 3, that many nucleotides, right? So if you multiply 135 by 3, so it becomes 405, right? So you have two options here which are close to it, 405, 408, and the others too far away. Now, whether it is going to be 405 or something else, how do you know? If you have read protein synthesis as a chapter in your molecular biology, you know that the first codon for any protein is initiation codon, which is AUG, right? Generated. Now, however, after the proteins are produced, then the first amino acid, methionine, it is trimmed by the peptidases to make the protein as per the genetic code, to eliminate the first one, which is not at all part of the genetic code. So therefore, you have 3 nucleotides which are required to initiate the protein synthesis in eukaryotes or otherwise prokaryotes. So methionine initiation codon. So if your codon is 405, you have to add 3 to account for the methionine, which is AUG. So it becomes 408. So correct answer is 408. So I cannot go on for analyzing all the questions because I don't have time, but I just gave some examples how one can go ahead with picking or choosing the correct options in the examinations. Here is a calculation again. The molarity of 15% sodium chloride is very easy. You have to convert the total amount present in a solution and align with the molarity calculation based on the molecular weight. So it is 2.56. Then here another enzyme kinetics question, but it is simple. In this question, you are saying that if your enzyme is 1 microgram per enzyme you are giving in the assay, as opposed to if you are giving 2 microgram per assay, nothing else is changing, only the substrate concentration you are increasing. So what is going to happen? The Vmax is going to increase, but Km is going to remain the same, and other options are automatically gone. So there are similar calculation questions, which is you can go through and you can find out the answers without much problem. So this kind of questions are there. I will move on to other kind of a little more complicated questions and then try to help you in answering those. During the annealing step in PCR, when the temperature is lowered, the reason why the original templates don't zip up with each other is, it is a very straightforward PCR question. Now, the question is, when say annealing step after denaturation, where the primers bind to the template, it is lowered, so why the original templates are not rejoining with each other. Instead, the primers get an opportunity to align with them through hydrogen bond. So the options which are given, best pairing is easy with the primers because of the short length, which cannot be correct. The concentration of the primer is higher than the concentration of the template. So it is a competition, and therefore this option obviously is the answer. The third you can eliminate, like long chain single-stranded DNA gets entangled, preventing them from base pairing, which cannot be true. the temperature of annealing is low enough for primer binding, but not for the template binding to each other, which cannot be true. So like that, if you read question carefully and use your thinking a little bit, it is very easy to crack the right option in these cases. There are questions like in genetic engineering, there are questions which can be easily solved by reading each of the chapters very carefully, and also your attention being paid to your classes when the teachers are teaching those topics. Now, there are many questions which can be analyzed. I think I will pass on these questions. As it is, this is going to be recorded and put in the ERP, so you will be able to see those, and taking guidance from this kind of examples, you can solve many questions from different years of previous questions from both CSR as well as DBT and so on. There are some questions from cell biology, like cell cycle, which is an interesting question. There are questions from intellectual property rights, which are taught to you. There are questions from physiology, questions from virology, then some questions from molecular biology, shRNA. Now, this kind of questions are also often set. You are given a picture, and then from the picture, you are asked a question. Like name of the blood vessel marked with a question mark, this one, in the figure below. Look at the options. Before looking at the option, pay attention to the picture given and think about the arrows. The arrows have some meaning in terms of your concept of artery and vein in particular. And this is the heart. So there are arteries are going out. There are veins which are coming in. Blood is brought to the different compartments, and then your blood is oxygenated blood is given to the lungs and other places. So if you have this looked at carefully, the answer becomes obvious. It cannot be cardiac vein, it cannot be cardiac artery. So these two options are gone. Now what remains is pulmonary artery and vein. The moment the arrow is going out, so it has to be artery. So therefore, B is the correct option. Now these are questions from the cell cycle. There are questions can be from immunology, many of them immunology questions. And I think I will stop at this. Basically, I gave you some clues as to how to arrive at the actual answer to the question based on the understanding on the question, reading the question carefully, looking at the keywords, looking at whether it is a negative question or a positive question, and looking at some of the obvious which are eliminated automatically, and therefore your option becomes reduced from 4 to 2 or 4 to 3, and that becomes easy to answer this question. So I'll stop here, and then I think we will have time for question and answer by the students. Was it clear? Yeah.
Yes.
So I think I have taken enough time. Now, if there are questions which should be read out by one of you, and then I will try to answer the questions.
Thank you, sir. I guess most of the students have a benefit from this, your guidance. And now I request Dr. Viniti to have some questions. Already, I guess, few of the questions are answered by the panelists already. So whatever questions are there, I will request Viniti ma'am to please read out the questions.
Yeah.
Yeah, thank you, Priyanka. And thank you, sir, for a very insightful session. I have few questions which are very specific. I'll just club 2 or 3 questions which students are requesting a mock test in the college, and most of them are saying that we can conduct these mock tests while we are at home due to the lockdown. So what can be done regarding it? Surely for students who have questions regarding mock tests, we do conduct regular mock tests in our college, and Amol sir would know better about it, but we have got very less response so far. So we are just hoping that we would get a really good response after this webinar. Yes, sir, over to you.
Okay, I think it is a routine thing that we have been doing for the last 2 years. As you mentioned that to begin with, the students are enthusiastic, but then somehow they do not turn up, at the day of the mock test. Now, if they are having a lot of time at home, so naturally during the second week of June, Amal sir can organize one kind of mock test with about 50 questions or so, and that is possible. We can conduct that.
Okay. Okay, then there is another question which is also similar to this. They are requesting for some college guidance on a regular basis regarding these entrance exams.
We generally do a workshop, no, 1-day workshop with 4 or 5 speakers. But this time, I don't know whether that would be possible. If we can organize online, we can think of organizing one again towards the second, third week of June or sometimes like that.
Okay. Yeah. And there is one interesting question. I think all panelists can answer this. So what are the exams which should be given for industrial jobs specifically, and which would lead them to good packages? So
Actually, I forgot to mention that there is a specific exam by Department of Biotechnology called Biotechnology Industrial Training Program, or in short, BITP. Now that examination is also online, followed by interviews. Generally, there are about 700 companies who like to recruit these DBT, through DBT, the biotechnology candidates, BTech or MTech or MSc. They have an online test, and followed by through online test, they screen about 1,000 or 1,200 students, and then they bring down the number to about 800, and then after all 800 students are called at different regional centers for interview, and then through an expert, and then they are selected, and based on their choice of industries which are given to them earlier, there is a matchmaking which is done by DBT, BCIL, and then they are given 10,000 rupees per month as stipend, and also 50,000 rupees to the company as bench charges, so that 6 months they can work there. And many a times, about 30% of these BITP trainees, they get absorbed in the respective companies if they do well, and they are very likely to be absorbed by the companies as regular employee. So this training, BITP, actually I have been associated with BITP for many years, and DBT-JRF also. BITP is a little easier test, even easier than GATE, okay? They are basically, that is made easier particularly because they are not going to go for PhD but for industry. So there is a little more industry-oriented questions and simpler questions, so that many people can qualify. Those who do not want to go for PhD and such kind of program and go for industry, there are easier kind of questions are made so that we can screen many of them, and then to interview, which is more important, to check their interest in the industry and their hands-on capabilities. So those 2 points are looked at in the interview, and then based on their marks obtained in the admission test and the interview put together, they are actually shortlisted for the placement. And that generally happens in June, and it's likely to be delayed this year particularly, I don't know. Okay. Yeah, so that is one government organized fellowship supported training program only for industrial activity or industrial placement.
Okay, thank you, sir. So I hope audience have taken note of this exam and the details.
Yeah.
And there are a lot of questions coming in regarding the study material, which should be used for competitive exams. So hard copies or PDFs or certain other links which they would be using for preparing.
Yeah. To me, I think the standard textbooks is the major requirement here. And there are no MEDGs or there are no specific textbooks for this kind of examinations. The best thing is to go for standard textbooks. You can have the soft copies and you can continuously concentrate on specific chapters. As I've said, you can choose the topics where you are more comfortable, or if you are more comfortable, you require less preparation for those topics, and then you concentrate on those which you are not so well informed, and from standard textbooks. And then you can also solve questions, practice solving questions. These are the 2 key requirements for qualifying these examinations.
Yeah, thank you, sir. So I think we have covered most of the important questions. Some of the questions, students, if we are not taking it for answering, it's already answered in the webinar. This would be live on our website as well, so you can go repeat with the webinar and check for your specific answers. And I think we have covered most of it. There are a lot of questions also regarding the GRE TOEFL exams, and while filling up the registration form, also students have kind of they are interested in GRE TOEFL. So I think we would in our next webinar we would give insights about that, or we can call someone who would be expert in that.
Yeah, I think the best idea is that I already indicated. We can look for a person or an expert, a couple of them, and we can have a workshop, 3-4 lectures in a day in the form of webinar that we can organize. So we have to identify some experts on this area, particularly GRE TOEFL and additional examinations and so on. So our group who are looking after these, particularly with Supriya, we can organize that and can discuss the feasibility of setting those programs sometimes in June or July. Okay?
Yeah, sure, sir. Thank you. So I think we are done with most of the questions. Priyanka, I'll hand over to you now. And for those who have still many questions which are unanswered, all the faculty members would be there. You can individually ask or email us regarding that. We would be definitely be answering those. Okay, so over to you, Priyanka.
Thank you, Vinidhi. So I hope most of the queries are solved. And one more query I wanted to solve here is students were asking whether to join a class for a GATE exam or not. Is the query of one student. So I just wanted to tell you that it is depends on how you prepare. So I'll tell you my case. I have applied for a GATE after my graduation, and that was like yes, all my friends are doing, so that's why I have again applied for the examination. And I went for the examinations, and obviously the result is that I got failed in the examination because I have not prepared anything. then after that, I met with one of my brother's friend, and he told me that these are the references and this is the way that you have to move. So then I started learning it, and then I've prepared the study material on my own self. And for 2.5 months continuously, I was doing the same thing. So it is not like that I went for some classes or all. What is required is just the dedication. If you want to clear some exam, you want a dedication, but if you want to go for a class, that is your choice at all. But we can clear it on our own as well. So only what is required is the right kind of study material and your dedication that will help you. So that's all from my side. So I hope this is a seminar, a webinar which has helped you, and I'll thank you all for joining with us. And one more thing, I'll ask you to all the participants to just, I'll request them to fill the form which will send you in some while. That is the form. You have a feedback form you have to fill, and after that we'll send you the participation certificate before the 10th of June. You'll receive your participation certificate on the mail ID which you have given through the registration. So that's all. So I'll thank you all, to all the panelists, to Pal sir, to Neelu ma'am, to Viniti ma'am, to Amol sir, to Gaurav sir for organizing this, and to all our faculties for doing this. So that's all. So we'll stop here.
Thank you very much. Thank you all. Thank you, Mr. Gaurav, for helping us with the webinar. And thank you, students, for participating in this. Keep at it. It is not difficult, and it is very important to have that acquired sometimes during your career. Independent of whichever career you choose, you can show on your credential that you have qualified one of these national competitive exams. That may be useful in the long run as well. Okay, thank you very much. Stay well, stay safe, and wish you all the best. Thank you.
Thank you all.
Yes, sir.
All.
Thanks, sir. Thanks. Nice session.
Thank you. Thank you.
Sorry, I went to sir. Sir came here, no? That's why.
Okay. Anyway, thank you very much for your help, and
Yes.
we will hopefully have some more in the second week of June.